Good evening, everyone. Thank you for being here today. It's my pleasure to present my topic of research, which is computational insights into the binding affinity of membrane protein-protein complexes and their mutational effects. I'm Fathima Ridha, doing my PhD under the guidance of Professor Michael Gromeha from Department of Biotechnology, IIT Madras, India. Let's dive into the topic with a quick introduction. Membrane proteins are a diverse class of proteins that resides in the cell membrane, and they make up around 20 to 30% of the human proteome. Their significance lies in their diverse functions as like transporters, receptors, signal transduction, etc. So as a result, they are primary drug target for nearly 60% of the drug in the market today. Numerous computational studies have been done on various aspects of membrane proteins, based on structure, function, topology, stability, mutations, etc. However, there is no large-scale study being done on the aspect of binding affinity. While if you see globular proteins, the binding affinity is still widely being studied. Most membrane proteins function as complexes, and to understand their function, one of the factors which dictates is the binding affinity. So what I mean by binding affinity is basically the strength of interaction between the two or more interacting protein molecules, which is quantified by the term delta G, which is the binding free energy given by the formula. So experimentally, this can be determined by various techniques. However, these are resource intensive and time consuming job. Also, it is important to understand the mutational effect because mutations are going to affect the interaction between these proteins, affect the binding affinity and impair the functions and ultimately lead to diseases. However, due to the intricate nature of the structure of membrane proteins, the binding affinity is still widely being not studied. Firstly, that there is a lack of readily available affinity data on membrane proteins in the literature today. Secondly, what are the factors of, influencing the affinity in membrane proteins is not completely being explored. Thirdly, there is no specific method for the binding affinity prediction for membrane proteins and their mutations. So that's the motivation for my work. So my first objective is to develop a database, which is called MPAC, which is a database specific for the binding affinity of membrane protein-protein complexes and their mutants. So as I said earlier, when we looked into the literature, there are data specific for membrane protein binding affinity, but they are rather scattered. So firstly, we need to compile these data into an effective, into a comprehensive database so that this can be used for further analysis. So that was the motivation for this objective. So we made the first database specific for the binding affinity of membrane protein complexes and their mutations. So we covered over, we looked into over 800 literature papers, and we collected data from PubMed. We collected all these data details like membrane protein complex details, experimental conditions, thermodynamic details, literature information, etc. And we also collected data from the existing databases, which are for protein-protein complexes in general, and we extracted the ones that are specific for membrane proteins and then manually collected all these details. So we developed this database. This is the homepage. So we have two different search options. Firstly, there is a simple search where you can just search by some keywords like protein, PDB ID, UniProt, etc. Also by entry ID. So this gives the complete details on whatever is related to, the contents related to that entry ID. Users can also do a quick advanced search, like you can query using a combination of multiple search options and get the results. You can download it in Excel form as well. We have option for downloading the entire database on request. So if you would need it, you can just feel free to fill in the request. And also, if you have some new data like specific to binding affinity of membrane protein complexes, which you feel is not included in our database, you can feel free to submit in this request form so that we will include that data. Because it's a database, it's constantly being updated. So currently we have nearly 5,400 entries, which includes both wild type and mutant data. And the next version will be released in Jan 2025. Okay, so coming to the second objective, so next is to make a ML-based prediction method for predicting the affinity of novel membrane protein-protein complexes. So as I said earlier, experimentally determining is a quite time-consuming and resource-intensive task. So you cannot do it for large-scale analysis. So that's where we need accurate computational method for predicting. So that's what the motivation for this work. So we developed a novel sequence and structure based ML method for predicting the binding affinity. So overall, we got a correlation of 0.83 and MA of 0.91 in jackknife test using 114 complexes. We also classified the complexes based on the membrane protein type, like single pass, multi-pass, as well as the functions. That actually helped to, we also saw good performance in each of the class. And the aromatic and charged residues at the interface and electrostatic interactions were found to be important in most of the classes. And on the independent blind test set, also we got a good correlation, ME, which is like less than 1.87. We also developed a web server for this. So if you know the PDB ID, you can put in the PDB ID and specify the interacting protein chains. Or if you have a model, like a new complex, which you want to predict the binding affinity, you can you can upload the file in the PDB format and select the protein class, whether it is a single pass or a multi-pass, and also specify the function of the protein. If it doesn't fall in any of the three, you can put it in miscellaneous. And so we will predict the delta G as well as the calculated KD from it. So now the third objective is now that we have made a server for the wild type prediction, we also need to look into the mutation prediction. So what is the change in the binding affinity upon mutation? That's what our third work does, which is MPA mutepred. So mutations in membrane proteins, they are culprits across broad range of human diseases, like from cancers, cardiovasculars, etc. So specifically, these are interface mutations, which are contributing to the diseases. So by changing the binding affinity. So we propose a novel delta G prediction method. So delta G is basically the change in binding affinity, which is delta G of the mutant minus delta G of the wild type. So here we combine the linear and non-linear ML algorithms to make a more robust model. So using our database, the data set used was from our database. We have around 856 mutations and 770 were used for training. And for the mutations, we calculated various structure and sequence-based features. And after pre-processing, we did forward feature selection. And feature selection, we used linear regression model because of for the better interpretability of the features. And for training, we use the nonlinear model, which is we use gradient boost regressive. So coming to the performance, so we have a 0.75 and 0.73, the correlation and ME in jackknife. And on the independent blind test set, we also have a good correlation and a lesser ME. So we also looked into what is the contribution of each of these features to the model prediction. So we did a SHAP analysis as well as a feature ablation study. And what we found was that the change in the overall stability, total energy, as well as the PSSM, which is a conservation-based feature, is important because when we remove that, the MA increased and the correlation decreased. We also compared our method with the existing, existing methods, which are for predicting the binding affinity of protein-protein complexes in general. So all these methods are for protein-protein complexes in general, and mostly it is for the globular proteins. So for our blind test set, we checked that these data are not included in any of their training or something, and we compared we evaluated the prediction. So we have a good prediction over the others. So this is a web server. So again, you put in the PDB ID or upload the PDB file and specify the mutation. We will predict the delta G, and also we will say whether it's decreasing or increasing the affinity. So, so coming to the conclusion, so we have made 3 computational resources for the binding affinity of membrane protein-protein complexes. So we anticipate that all these resources will help contribute to enhance the understanding of membrane proteins and complexes, because there is no much study being done in this aspect. So this can help further into diverse analysis, and also in future help in drug design. So yeah, So these are the publications. These are the QR codes for my servers. If you want to use, you can just like scan it. Yeah. And lastly, I would like to thank my guide, Professor Michael Gromiha, my institute, my lab, Protein Bioinformatics Lab, and PMR for the scholarship, and last but not the least, the conference APBJC for inviting me to the talk. Thank you.
Thank you very much. So now session is open for the question and discussion. Is there any question or comment? Okay. Maybe this is a very naive question, but you develop interaction energy delta G prediction method for the membrane protein complex. And maybe the protein interface is not much different from the soluble protein in the membrane protein. So what happens if you apply your method to predict the delta G or delta delta G protein of a soluble complex protein? Did you ever?
So, okay, you're saying I apply my method for the globular proteins. What happens? I I haven't checked that. Like,
Okay.
but I think the performance will not be so good because the features we have is a unique combination of features specific for like membrane proteins. So the features is defining the model, right? So the combination of features is contributing to the model prediction. So prediction performance for globular proteins will not be so good because this is
Okay.
specific for membrane proteins.
Okay. Is there? Maybe the last 2 or 3 slides, you compare your method performance with other existing method. May I see that
Yeah.
again? Yes, it's wrong. I don't know much about this method. This method compared to yours specifically predicts the structure stability of the membrane proteins. For example, the MCSM PPI2 is specific for the membrane protein.
No, these are not for membrane proteins. These are for protein-protein complexes in general. So it includes membrane, protein, globular protein, all the proteins.
Oh,
So there is no method specific for membrane proteins, so that's what we have developed.
okay, I see.